Welcome to AP Biology. Today's video is going to be on the control of prokaryotic genes, how bacteria basically regulate their gene expression. So bacteria have a slick thing that they do. In their genome, if they need, for example, 3 enzymes to break down a particular sugar, they will put all 3 genes for those enzymes back to back to back. So then they can just create one situation where the RNA polymerase is cruising down the DNA copying genes, but they'll copy 1, 2, 3 genes right in a row. And they'll make 3 messenger RNAs right in a row, which will lead to 3 proteins. All those proteins will end up being enzymes that are needed to break. So they make everything needed for a particular job all at once. That's different from how eukaryotic organisms have their genes set up. So basically what you'll see is enzyme 1, 2, 3, whatever, all of the enzymes right in a row, or all the genes right in a row to make these enzymes so that they can do whatever function that they need to do with those enzymes. We call this sequence of genes that are all right in a row, we call that an operon. And the operon includes a couple other things too. But the big thing is the genes being sequenced all right in a row. So in an operon, we have what's called prog. So you have a promoter region, a repressor operator situation, and then all the genes right in a row. And these operons can either be inducible operons, which are operons that can be turned on, or repressible operons, which can be turned off, and we'll talk a lot about that in our next video. So here's the basic setup of a operon. You have your promoter region on the DNA. That's where the RNA polymerase is going to attach to. And then you have your operator region. That's where the repressor is going to attach if it's repressing. And it will prevent the RNA polymerase from copying the genes. And then following that, you will have your genes that are needed to make the particular enzymes that are going to be used to do a particular job. So that's our operon model, and we always refer to it as prog, promoter, repressor, operator, and genes.